Welcome back to studying Prescott's Microbiology. All the Homo sapiens are welcome. And wait. Before starting to study, we have to correct one thing, and that is our chat. So give me a second. I need to correct the chat first. Because without that I can't see if someone texted or if someone messaged me or if someone do whatever. So let's turn this on. My channel needs to be on. So even though we are still not starting the study, I'll revise what we studied till now, right? Wait. We don't want disasters. See the small thing here at the end here. This is the live channel portion or whatever. So it's not working. It say the live chat is currently unavailable. So I'm going and correcting that. So last time, by the way, it is after quite some time since I streamed. The gaps are getting bigger and bigger every time. Oh wait. Oh, holy. It's supposed to be public. Yeah. Oh, so let me start because it was not public. Wait. Here. Hello again, Homo sapiens. Welcome back, studying Prescott Microbiology, and of course we are not playing. These are some cherry blossom for you, and now, yep, we are stopping and we are starting study. But before that, I am just searching for not searching, I'm just trying to see my live chat, because if I don't have that, I won't be able to work properly. So I need to pop out that first. I need to text me so I can see if it works or not. So pop out that. I need to pin it. Don't worry, we will be studying. I mean, no one is that excited for studying, right? So Control+C. I don't like the guidelines lines, so there is that too. And now our OBS, so I can see. Oh no. Control+C. Okay, so I need to change this and see if it will work. I just hope it works. Oh, okay, it works. It is great that it works. So why did I turn this off again? Also, we need to, as soon as I close this and this, we are turning on the eye, so in case that we study. Okay, let me do high more, so this new guideline thing go away. A, B, C, whatever it is, I want this to go away. You know, this welcome to chat and stuff like that. I need those to go away and actually see if the live actually works or not. I think I should cut my chats a little bit more, you know, like a little bit more, since I think it's taking up even more space. Half of my screen is actually just chat. So should I cut it more? I mean, it's fine, I guess. It's not fine though. Whatever, it's fine. We are starting the stream. We don't have time for this stress. So today I changed two things. First, the avatar voice was first, like minus to it was sharp, and I noticed that it was not very positive because it was not understandable. The next thing I changed was is I will be reading it. Before, when I started this channel, I used to read it and then search the Google Midway, but then I changed it and started studying before the stream, so the stream is not getting 1 hour, or it was, the time was getting too much, but it was started to stop having fun for me, right? And I started getting bored, and so the streams, obviously I'm busy, but I also had no stamina to do that. So I am going to read just as I used to, and will search the Google Midway, because I want to enjoy reading it rather than try to teach it. So yeah, anyone on that is, if anyone wants to study and see me struggle understanding this, then they are welcome. They can learn things with me, or they cannot, or they can study from the professors or teachers if they are in a hurry to learn, because my purpose is just to gain knowledge and enjoyment. So anyway, that was for quite a long intro. And after the intro, we are going to come back to our topic, and we will have fun studying, because I don't plan to be boring, or because I myself don't enjoy that. I have noticed myself doing that. So the first thing is chemotaxis. So from what I know, this word, the chemotaxis, is wait. What happened? I hate when this occurs. So it's going to be 80. Am I on the right page, or make it 90, I guess? Yeah, it's not that far now. Last time we were studying these, by the way, this was such an interesting topic as well, but I taught it in such a boring way. I don't even know if someone will be understanding it or not. But if my mic is low, always tell me, because I don't hear myself. Anyway, from what I know is chemotaxis. Chemo is from chemicals, right? The chemical, and taxis means it is moving. Tactic. It came from tactic, or you can say taxis. So it is the movement which is done because of the chemicals, because of the presence of chemicals or effects of chemicals, or bacteria. Some bacteria are motile, they move. Not all of the bacteria are motile, but some are. And even if some are not motile, they grow in a way that they move in the direction of where the chemicals are they want, or the chemicals that they want to run away from. They start growing on the side that which they want. Like, for example, if I tell you this, for example, this is sugar. Wait. For example, this is sugar, right? These are bacteria, colonies. They are small, but the bacteria will start to grow onto the sugar side if they like sugar. Usually they like sugar because it's glucose. But in case there is HCl, consider there is a strong acid chemical. And if you put the bacteria small number here, they will, even if they are non-motile, they will start to grow on this side. They won't have growth on this side. But again, this is the growth, so this is not like chemotaxis, because they are not actually moving, right? It will be at chemotaxis when they are actually here, and then this bacteria try to move this way or that way. In case of sugars, of course, this way, of course. I don't think there is any bacteria that doesn't like glucose, and if there is, that is extremely interesting thing. So bacteria do not always move aimlessly, but are attracted by such nutrients as sugars and amino acids and are repelled by many harmful substances and bacterial waste products. So yeah, this is what I was blabbering till now, right? They are attracted by some stuff and they are repelled by some stuff. Bacteria also can respond to other environment cues such as temperature. Okay, so temperature is not chemical, so that's why it's not chemotaxis. But the response, they are talking about a response, not just movement. So there is that thermotaxis, and then for light phototoxis, oxygen aerotoxis, osmotic pressure osmotaxis, gravity, gravitaxis? They didn't include the word for gravity. Why? Did we lack vocabulary? I mean, we can always start making words. So that means that there is not just chemotaxis, there are a lot of different types of taxes, right? But what we are studying will be chemotaxis, and what we are studying will be chemicals. The movement towards chemical attractants or away from repellents is known as chemotaxis. So it's all about movements and chemicals. Chemicals can be attractants and repellents. Either they attract bacteria or either they repel bacteria. Such behavior is of obvious advantages to bacteria. Advantages in that way is that they are able to safely go into the environment that they want to, right? Now, chemotaxis may be demonstrated by observing bacteria in chemical gradients produced when a thin capillary tube is filled with an attractant and lowered into a bacterial suspension. They are saying that chemotaxis may be demonstrated by observing bacteria in the chemical gradient produced when a thin capillary tube is filled with an attractant and lowered in a bacterial suspension. So this is the practical way. You take a capillary tube. We all know what capillary tube looks like. It's like simple tube but has capillaries, veins in it, right? So even if we think we know, we should just search it. How does it look like? Capillaries tube. So this is how it looks like. Oh, they are so thin. Actually so thin. It's more like an injection and not tube. Okay, so they are capillary tubes because they are like small capillaries instead of a big place having a lot of space. As attractant diffused from end of the capillaries way, let us draw while they are teaching us. This is a capillary tube. What is this capillary tube? Anyway, it is capillary tube. It is very thin, which I drew very thick. It is filled with an attractant. Now it is filled with an attractant. You know, it is filled. This has an attractant. This has a chemical in it. And now we have a beaker or a bacterial, anything that has bacterial suspension. And now it is dipped with that. It is lowered into that. As the attractant, okay, so this area, this end of the capillary tube, it can diffuse this chemical. So from this area, the chemical starts seeping out. It diffuses, and the bacteria collect and swim up the tube. Now bacteria that were present here and there and scattered, they start swimming up and going towards this capillary tube. Now the number of, and not just outside, they start moving inside because they think, oh, if the end has a lot of product, we will have more product on the inside as well. So they say the number of bacteria within capillaries after a short length of time reflects the strength of attraction and rate of chemotaxis. So you will be counting the number of bacteria inside the capillary tube. So the more the number is, The higher the number is, the higher the chemotaxis that occur, and the higher the chemical is attractant. And if you see the bacteria moving away from these areas, that obviously means that it is a repellent. So the one that the bacteria moves in are the one where the bacteria moves in are the positive chemotaxis, and the one where it aways from it, it is the negative chemotaxis. And they are saying that the positive and negative chemotaxis can also be studied by Petri dish. If the bacteria are placed in the center of dish of a semi-solid agar containing and okay, so this is Petri dish. Wait, this is not. If this is a Petri dish, consider this a Petri dish. And we put bacteria in the center of it. This is bacteria. And this is semi-solid agar, means it is a jelly-like, not completely solid and not completely liquid. Why is it not completely liquid and completely solid? Because if it was completely solid, bacteria won't be able to move it across it. But because it is liquid, they can move. Containing an attractant. Let's say the attractant is placed here. The bacteria will exhaust the local supply and then swim outwards, following the attractant gradient they have created. So in this center, of course, we have nutrients all around the Petri dish, right? All around the Petri dish and in the same amount. Now, if after bacteria complete these areas of nutrients, they can move to this side, but they won't move to this side because these nutrients are low in comparison to this one, so they will automatically move to this area. So after they exhaust this local supply, they will swim outwards, following the attractant gradient. The result is an expanding ring of a bacteria. Expanding ring of the bacteria. When the disc of a repellent is placed in a Petri dish of the semi-solid agar, the bacteria will swim away from there. Like in case this area was repellent, the bacteria will all start to form rings in this area. The growth will show on this side rather than that side, even if the nutrients are low on this side. I mean, we are talking about in comparison to the attractant. Creating a clear zone around the disc. Oh, okay, so this type of thing is the chemotaxis one. It is the movement, it is okay. So I showed the movement one. What they are talking about is not the movement one. They are just saying put the bacteria here and put the chemical attractant here as well. So if they like the chemical attractant, they will form a ring around it, and if they don't like the bacteria, there will be bacteria. The disc will have empty areas around it, will have no bacteria. Means if we put two discs of chemical repellents, so automatically bacteria will grow on other areas but not on these two areas. While if we put multiple discs of chemical attractants, we will see all the rings around this because it will show growth. So they talked about the disc type, but usually when we talk chemotaxis, we talk in this style. So yeah, well, I didn't know this was chemotaxis because it didn't involve movement. It just showed the response to either it will grow or not. It's more like if they like the chemical or not. So yeah, I don't know where is the taxis part in that. So the bacteria will swim away from the repellent, creating a clear zone around the disc. Bacteria can respond to very low levels of attractants, about 10 raised to power minus 8 for some sugars. Low level, I said they like the sugar. So even if they are in low number, they will still like it because they are sensitive to it. The magnitude of their response increasing with an attractant attractant concentration. So they are saying the magnitude of their response is directly proportional to the attractant concentration. The more the amount is, the more their response will be. Usually they sense repellents only a higher in concentration. They say usually it's obvious. The more it is, the more easy for them to show response to and to notice it. The lesser the amount is, they might notice it, but it doesn't sense. They don't sense it. Repellents are only at higher concentration. If an attractant and a repellent, okay, so they are talking about only repellent. They say the repellent, that the attractant can be sensed even in low amount, but the repellent is attracted only in higher amount, is only sensed at higher amount. So it's like they are blind to the repellent. If the attractant and repellent is present, the bacterium will compare both signals and response to the chemical with most effective concentration. Oh, I see. So it's like this. For example, this is petri dish. This is bacteria. We have at this site low amount of attractants but high amount of repellents. Okay, so this is a cross one, okay, and this is the good one. This is the negative one, bad ones, and this is the good one. But bacteria, we know that they sense things in higher concentration, right? So in case if you put two things together, even though they sense this, but the response they will show towards is the repellent, right? Yeah, with the most effective concentration. So that means they will respond to this in a way that they will be moving away from it. It's not because they notice that there is an attractant present here. It's because they notice that there is a repellent there, that's why they will be moving on to that side. But in case both have the same concentration, right, then obviously bacteria again are going to that side, because they like to go towards the attractant. So, and in this case as well, if the good one has high amount and the bad one has small amount, right? The bad one has small amount, they will again obviously go towards this. Obviously two reasons, because one, the concentration is high, second, it is an attractant. The third scenario can be if the good one amount is small too and the bad one amount is small too. And now bacteria cannot sense this, because we have said that repellents are only sensed in higher amounts. So bacteria don't even know this in small amount, but they do sense the good one, the attractant, in small amount. So again they will be moving towards that side. So it's like they sense one thing or not, they will always be moving away from repellent and towards the attractant. So that's their kind of, Come on. I haven't noticed how these things work till now. Close. Attractants and repellents are detected by chemoreceptors. Now, how do they even detect it or sense it? They have receptors for that. The special proteins that binds chemicals and transmit signals to the other components of chemosensing system. So they receive it and then transmit the signal to their own systems inside them. Chemosensing, if one receptor is activated, and obviously other receptors are co-activated with that. About 20 attractant receptor and 10 chemoreceptors for repellents have been discovered thus far. This is very low in number. No, like I was actually ready for them to have hundreds of them, but they only have discovered 20 attractant receptors and 10 repellent. That's really low in number. So that's why they can't sense repellents in low concentration, because they have low receptors of that. These chemoreceptor proteins may be located in the periplasmic space of the plasma membrane. Maybe, okay, maybe, they don't know. So other than the chemoreceptors that are, well, we already said that receptors, receptors are usually on the cell wall or in the cell membrane. So they are talking about that these proteins might be on the plasma membrane or might be in the periplasmic space. Some receptors participate in initial stages of the sugar transport into the cell. Okay, so it doesn't mean that the receptors are only for sensing them. Receptors can work in transportation as well. So it's like receptor is not for one thing but can be used for multiple things. The chemotactic behavior of bacteria has been studied using tracking microscope, a microscope with a moving stage that automatically keeps an individual bacterium in view. So it's like a camera that follows only one thing that it's targeting. So in the same case with the microscope, it only targets a single bacterium that we have been that we fixed it on a tracking microscope. In the absence of a chemical gradient, E. coli and other bacteria move randomly because E. coli are not motile. Are they motile? Well, they do have flagella. That means that they are motile. So they are motile. For a few seconds, the bacterium will travel in straight or slightly curved line is called a run. So they are running if there is nothing present. When a bacterium is running, its flagella are organized into coordinate corkscrew-shaped bundle. We already studied how the flagella works, so they are just summarizing it for us. Then the flagella fly apart and the bacteria will stop and tumble. So it's like they are moving. They are telling us how they run and how they stop. The tumble results in the random reorientation of bacteria so that it often is facing in a different direction. They are just moving here and there until they can, just in a search of maybe food or whatever, until they sense something. Therefore, when it begins the next run, it usually goes into a different direction. Those who haven't studied the previous topic, I think they will have difficulty understanding it. In contrast, when the bacterium is exposed to an attractant, it tumbles less frequently, means it stops less frequently or has long run when traveling to towards the attractant. So it sensed something in that direction, and now it has the motivation to reach there, so that it will signal the cell to move and no need to stop. They stop because they have no purpose. So that's why the runs are not long, but in this case it is longer when traveling towards the attractant. Although the tumbles can still orient the bacterium away from the attractant. Yeah, when they stop, it doesn't mean the direction of their face, or they can say they will be facing towards the attractant. It can be something, but they will reach there over time. Over time, the bacteria gets closer and closer to the attractant. The opposite response occurs with a repellent. Yeah, they will just be saying run with either 4 legs or 24 legs when they sense repellents. Like, of course, it's a danger to them and their lives are in danger, so they will also show longer runs. Run time lengthens. See, again, the tumbling frequency decreases and the run time lengthens because they just want to get away from the area where the repellents are present. So when the bacteria moves away from the repellent, so both in attractant and repellent case, the bacteria runs faster and longer without tumbling. Now, positive bacterial chemotaxis. Okay, so they have shown colony of a motile but non-chemotactic bacteria, colony of a non-motile bacteria. Motile but non-chemotactic bacteria. Oh. I mean, it's, what is this one? Chemotactic motile bacteria. So you see the bacteria has moved towards the attractant because it is motile. Again, chemotactic ones are only motile bacteria. I don't think non-motiles will be able to move or do the tactics. So if you see they are motile but they are non-chemotactic, means they can sense chemicals. So they will be making colonies but not in that much high amount, because the surrounding nutrients are just, a normal and not so special material, so they will be going on a normal phase. Well, these one are non-motile, so they usually don't move and stay in one place, so their growth is slow because they are so close to one another. So the growth of other bacteria needs space and they don't have that space, so that's why they slowly expand. While the motile ones move, wait, let me show it with a diagram. I don't know how you guys will understand. You see, these are the motile bacteria. This is how we put them, and after few hours or whatever their incubation, no, not incubation, their log phase or growth phase, whatever. So when they are motile, they move. One bacteria will go here, one will go here, randomly, right? So the colony will get bigger. The next time you will see them, these spaces will be filled by other bacteria, so the colony gets bigger. While in the non-motile, it just stays here. It just stays, and the other one grows, and slowly they will make colonies. So it's like they are not taking up space. They don't take up space. That's why their colonies are compact and small, while these ones takes more space. Now in the case of these ones, now it will be the same case with the motile one, but again their run moves faster. They run for longer period and they move away, and just trying to reach that attractant. So they are in very large space, and again the nutrients, of course there is a reason they are attracted to that chemical. That chemical will obviously be giving them something, so that's why when they grow in high amount and in larger area. So let's see what they say. Chemotaxis can be demonstrated on agar plate that contains various nutrients. Positive chemotaxis by E. coli on the left. This is left. The outer ring is composed of bacteria consuming the serine. Oh, so this is ring. Okay, so the bacteria are surrounding the chemical. The second ring is formed by E. coli consuming aspartate, a less powerful attractant. I don't even know what they are talking about. Is this the first ring? Is this light one the first ring? This one the second ring? A less powerful attractant. The upper right colony is composed of motile but non-chemotactic nutrients. The bottom right colony is formed by... We understand these two, but what about these ones? One ring. Okay, so the upper ring, the outer ring. So the outer ring means the less obvious one is the serine. Serine or serine, I think. Serine, no, serine. And this one's aspartate. What about this one in the middle? Maybe it's just the chemical. I didn't understand, but whatever. We'll move forward. Maybe try to understand. Oh, okay. I see. So these white things are the chemicals, just as seen in here. So the bacteria are all around the media on the plate, but we put small discs and that are repellent. So when bacteria grows, they avoid them, move away from them, and form circles around them. So negative chemotaxis by E. coli in response to the repellent acetate The bright discs are plugs of concentrated agar containing acetate that have been placed in dilute agar incubated with E. coli. Acetate concentration increased from 0 to top right to 3 m at the top left. Note the increasing size of the bacteria-free zones with increasing acetate. Top right. Acetate increased from 0 to 0 at the top right. Okay, so here it's 3 mole. It's 3 mole. Okay, so this is right and this is left. At this, the concentration of which one? The concentration of acetate is 0, while here the concentration of acetate is 3 mole. So the higher the amount of the repellent is, the further away the bacteria are. So it matters on the concentration, because we have seen that the bacteria can't sense repellents in lower concentration. So that's why you obviously see the difference when the amount gets higher. The bacteria have migrated for 30 minutes, and this happened only in 30 minutes. Okay, while this one, tumble, run, tumble, run, tumble, run, tumble, tumble, tumble. Okay, see, if they were able to move in a straight line, they would have moved it very faster. But this one, look, it is moving in so difficult, complex movement, because it just don't know how to face it. It always face in a different direction. So even if it's trying to move forward, it goes on a long way. Oh, and the first one is random because it doesn't know where to go, while the second one, it knows where to go, and the tumbling frequency is reduced. So this is just the comparison of seeing that, see, they are moving quite accurately, and there is not that much of a difference. Okay, let me check something. Okay. View my channel. Give me a minute or maybe second. I have to check if the video is public or not. Otherwise, oh, okay, it is working. Current bitrate is lower than recommended. What is bitrate? Man, I don't know what is bitrate. The stream's current bitrate. Oh, so they are saying my net is low? Is this what they are saying? Okay, but just stop. We need to move forward. Now this was Chemotaxis. Although this was interesting to see how bacteria move in response to chemicals, and this is why they have motility. There is one reason that they are motile, and also we have learned different types. You know, the phototactic, the key. No. See, my fish memory. Thermotaxis, aerotaxis, osmotaxis, and I don't know what for gravity, gravivotaxis or whatever. So these are different types of tactics, movement, then rather different than chemotactic, right? Now, chemotaxis aside, wait, it is not done. Clearly, the bacteria must have some mechanism for sensing. Then it is getting closer to the attractant. or is moving away from the repellent. The behavior of the bacterium is shaped by the temporal changes in the chemical concentration. We have already studied this, that the mechanism for sensing is that it is getting closer to the attractant, and the mechanism for repellent is that it is moving away from the repellent. And again, the behavior of the bacteria is shaped by the temporal means temporary changes in the chemical concentration. Also, the bacteria moves towards the attractant because it senses that the concentration of the attractant is increasing. Attractants don't move, so why the concentration of the attractant increases? Probably because they start to sense it more and more over time. Now likewise, it moves away from the repellent because it senses that the concentration of repellent is decreasing. Okay, it's not that. I understand. See, this is a petri dish. Again, another petri dish. This is attractant. Now, for example, there is a bacteria. Again, this is the attractant, so it's a good thing. Now, bacteria tries to move towards it. Now, how are they going to sense that if they are moving in the right direction or not? So, when bacteria moves in this direction, for example, whatever tactics they take and they reach here. So, when they were here, they had less amount of signals that they were receiving through receptors. But when they reach here, obviously the attractant is closer to it, so there is more signals here, and the amount of signals increases, so they are satisfied, then they know that they are moving in the right direction. So that's why they continue moving in that direction, right? So this is how the chemotaxis towards the attractant works, right? Like this is how they satisfy themselves and knows that they are moving in the right direction. While in case of repellent, in case the bacteria is here and the repellent is here, obviously it's not a good thing, so we cross it. Now bacteria, as soon as they take signals from here, they will think, oh, we are in bad position, we need to move away from here. So they start chemotaxis, they start moving. Now when they reach here, obviously there is less signal. Just consider there are 3 chemical particles here, and they just take up 3 signals, while there they picked up 30 signals. So they are satisfied that they are moving in a right direction. They will continue to move until they reach an area where there is no signal for that repellent. Then they will stop moving. So this is the kind of mechanism. Okay, I see. This is how they set themselves that they are doing right thing. So the bacteria's chemoreceptors play a critical role in the process, obviously because they sense it. The molecular events that enable the bacteria cells to sense a chemical and respond appropriately are presented in chapter 8. Okay, they are saying it's not your time to study that. So the coritics, okay, now the chemotaxis is done. So we don't need to summarize it, I think, yeah, let's move forward. Bacterial endospores, how much is it? 1, 2, 3, 4, 5. Oh my gosh, too much knowledge, too much study. But bacterial endospores are very, very important in the bacterial life cycle. Just as fungi produce spores in propagation, they can survive in a lot of extreme conditions. So the same case needs to be done with bacteria. Fungi are actually multiple cells, but in comparison to that, bacteria have only one cell. They need to protect themselves in one way or another way from extreme conditions. So they develop the mechanism of endospore. I don't think there is any other mechanism that can let the bacteria survive for next generation other than the endospores, right? And endospores bacteria are the problematic bacteria, because even if we try to kill them and their endospores remains, they don't die. So now, up till now, the ones that we studied too much are the Bacillus, Clostridiums, and Sporosarcina. There are a lot of it, but Bacillus is very common when you say endospore. So, endospore, we know what spore is, right? No, we don't know. We go and search it. I know it in my mind, but we go and search. I think spore is a cell that has all the information present in it. Biological that allow fungi to be reproduced. Yeah, it is a cell that has all the genetic material and that allows the cell to reproduce, to continue the generation. So I think that's the general definition of that. Now, what is endospore? Now, the spores that the fungi produces are the outer spores. It is made from a special cells, and they divide and redivide or do mitosis, meiosis, whatever. So they form the spores. Those spores are not endospores. Endospores are the spores that cells make out of themselves. This is a bacteria. In case of fungi, they have too many cells. They will just transform, or whatever they have reproductive organ, they will transform this one cell and make the spore. The rest of them is surviving, right? The rest of the fungi is alive and thriving and surviving. But this one area, this will become spores, and this will be continued for next generation. This spore will go and make other fungi. But in case of endospores, and also fungi, even though produce spores, they do it even when they are not in extreme condition. I mean, bacteria don't do it, but they do it for the extreme condition. So in case of bacteria, this is the bacterial cell. This is completely normal. This is cytoplasm and this is their genetic material, right? But this is not enough. What if they face an extreme condition? How are they going to survive? So they do what? The cytoplasm that has a lot of water content, they say bye-bye to water, and the H2O is gone out of the cell. You won't see H2O in these bacterial cells. So it kind of shrinks. It's like you put a vacuum on the bacteria, and instead of air, you put out all of the water quantity of that. So the water activity level of the bacteria is very low, becomes very low, and then they form an envelope type of things outside their cell walls or anything. So it's kind of, they make an extreme hard shell around them, shell type of thing, and the inside water and stuff there is removed. Now also they take up a lot of nutrients and store in it. So in case if the bacteria needs to survive again and needs to reproduce again, it has that, nutrients, etc. So this is my understanding. Now let's see if I'm wrong or right, or how much I'm right. Obviously not by that much, because a number of gram-positive bacteria can form special resistant dormant structure. Again, endospores are dormant, okay? So they don't usually, try to live unless, what do you call it? Whatever. You won't be seeing them multiply under the extreme condition. It's like, we just existing, and we don't want to trouble anyone. Unless you give us favorable condition, then we are your enemies. So it's kind of that dormant structure. Endospores develop within vegetative bacterial cells of the several genres. Vegetative bacterial cells, right? Within the vegetative bacterial cells. Wait. So the endospore that I made was what? Oh my gosh. Inside. What? Oh yeah. I'm not wrong. I totally thought that I was going in the wrong direction. Yeah, within this threw me off, because they said within the vegetative bacterial cell. I thought that the bacterial cell itself becomes endospore, and they are saying that it becomes inside the vegetative cell. So it kind of the merged thing. Obviously, the remaining vegetative bacterial cell from which the endospore is formed also becomes a part of endospore. Remember, also becomes. So in short, it sacrifices itself for the endospore. I mean, it is an endospore in the end. So bacilli, clostridium, and Sporosarcina and others. The structures are extraordinarily resistant to environmental stress such as heat, ultraviolet radiations, gamma radiations, chemical disinfectants, and desiccation. Remember these things because it stresses the bacteria. Next time you see them, you can kill them with that. In fact, some endospores have remained viable for around 100,000 years. Okay, stop. I wasn't thinking that far away when they are saying that they can survive in harsh conditions and stuff. 100,000 years? How many decades are they? No, I mean centuries. How many years are in 1 century? 1 century is how much? 1 lakh? No, wait, 100 years? How many centuries are 100? 1, 2, 3 years. No, like, are they kidding me? Did it take 100,000 years for them to have a favorable condition? 1,000 centuries? 1,000 centuries. Oh, okay. So they survived for 1,000 centuries. Centuries are cool, that's why I'm doing that. Because of, come on, the ad. They don't even leave me when I'm studying. Okay. So because of their resistance and fact that several species of endospore forming bacteria are dangerous pathogens, endospores are of great practical importance in food industries and medical microbiology. Why is it always the pathogenic ones that they have the mechanisms for surviving? Like, why can't it be the good ones that can form endospores? Or maybe there are, but we are just focusing on the ones that, endospore forming have dangerous pathogens. Bacillus species, genre is not usually a dangerous species, dangerous genre, right? Bacillus. They have mostly, I think they have 95% of the bacteria are great, like not harmful at all. But there are some that are harmful, and that are maybe endospore forming as well. So this is because it is essential to be able to sterilize solution and solid subjects. So why is it important? Because we want to get rid of the bacteria. So if we didn't know that the bacteria can form endospores, how are we going to get rid of them completely? Now that we know that they form endospores, we can proceed with the what do you call, with the right procedures to get rid of them. So let's see how much they can handle the sterilization techniques. So endospores often survive boiling for an hour or more, therefore autoclaves must be used to sterilize many materials. So they say for an hour or more, 1 hour, 2 or 3 hour, man, we can survive. So therefore autoclaves must be used to sterilize many materials. So even because boiling just makes the temperature only 100 degrees centigrade, right? But the autoclave makes the temperature 121 degrees centigrade. Even though the difference doesn't look that much intense, it is intense for the bacteria. Even 1 degree centigrade change in the temperature is way too dangerous for the bacterial growth. Just change the optimum temperature of the bacteria by 1 degree centigrade and you will see impact of them growing. So automatically, if they can survive in 100 degrees centigrade for hours or so, they can't survive in autoclave for even 15 minutes because the temperature is so high. Usually we keep that at 121 degrees centigrade. Endospores are also, because the autoclaves use steam. Steam is way more damaging than your general heat, dry heat. So because steam, first it is water, so hydrogen bonding. The second, it is extremely high temperature, so the molecules are extremely energetic and they start and meddling with the bacterial or their own cell walls and membranes and stuff, so it kind of degrades them in different way. Also, the atmospheric pressure becomes so high that the bacteria can't survive, that it bursts. I think so. Endospores are also of considerable theoretical interest. Now, why is it theoretical interest? Like, what do they want to study in that? Because bacteria manufacture these intricate structure in a very organized fashion over a period of few hours. Spore formation. What? They form the endospores that can survive for 100,000 years in few hours. Ah, wow, few hours are enough for them. What do we do in few hours? Sleep. Spore formation is well suited for research on the construction of complex biological structure. In the environment, endospores aid in survival when moisture or nutrient are scarce. Yeah, so their stressful environments are not just the UV light, the gamma radiations, the chemical stress. It is also the nutrient depletion and the moisture deficiency or extreme humidity. I mean, they all have optimum things, optimum temperature, optimum water levels, optimum nutrition for them to grow. Here and there changes in that causes stress for them. They are very sensitive and picky about their surroundings. Endospores can be examined with both light and electron microscope because endospores are impermeable to most strains. They often are seen as colorless areas in the bacteria treated with methylene blue and other simple strains. Special endospore stains are used to make them clearly visible. So first thing first. Here we know that we do gram staining and they are visible because all of them are divided into two classes, either gram positive and gram negative. Maybe except mycoplasma because it doesn't have even a cell wall. So they are saying that they are impermeable. Means how do stains work? Stain needs permeability because it diffuses into their membranes and stick with some materials. That's how stain works. So that's why it is colorless in most of the strains that are methylene blue in case of what do you call it? Wait, crystal violet is what? Methylene blue? Colorless areas in bacteria are treated with methylene blue. What do they mean by colorless areas in bacteria treated with methylene blue? Another simple strain. Special endospore strains are used to make them. Oh? Yeah. Endospore positions in the mother cells, sporangium, frequently. Okay, so when they are talking, probably they are talking about the earlier stages of the endospore formation, where it is like this. It is like this when we stain the bacteria, even though the stain goes inside, these areas are seen as colorless. Man, I don't understand what they are saying. Colorless areas inside the bacteria? Colorless inside the bacteria. Is it this one? Oh, maybe. Look at this one. Oh, I see. No, what I see? I see nothing. Is it this one? No, like what is official? IFM? C? Cytoplasm? Man, let's leave it. We understand that after we study the life cycle. Where is it? Endospores. Okay. Endospore. Okay, so they said it needs special stains for us to see the endospores. Endospore position in the mother cell, sporangium, frequently differs among species, making it of a considerable value in identification. Oh, so it helps in identification of the bacterial species or strains or class or family or whatever, because usually in microscopy, even though we see bacteria From the color we can say that this is gram positive and gram negative. From the flagella we can say it's motile or non-motile, etc. We can understand this few stuff, but it doesn't help completely in identification of the bacteria. So if we have endospore, it actually complexify the microscopy study and actually helps in understand for the biomedical for the biochemical analysis. I mean, you still have to go through it, but it is still helpful to understand, oh yeah, so this may be in that class, because first it has endospores, second the endospore looks a little bit like this and a little bit like that. Endospores may be centrally located close to one end, subterminal, or definitely terminal. Sometimes an endospore is so large that it swells the sporangium. Okay, so first thing is the endospore is in the cell. First thing first is that. So sometimes it's large, sometimes it's small, and sometimes it is at the sides, usually close to one end. So it can be in the center, it can be at the terminal region. Usually they are terminal. Now, electron micrographs show that the endospore structure is complex. Okay, why is it complex? The spore often is surrounded by a thin, delicate covering called exosporium. A delicate covering. Okay, so the first covering is the exosporium, and it is a thin, delicate layer. A spore cord lies beneath the exosporium. Usually the thin layer should be inside while the thick is out, but in their case, thin is outside and the coat is inside. Beneath the exosporium is composed of several protein layers and many fairly thick. It is impermeable to many toxic materials and is responsible for the spore's resistance to chemicals. The core also is thought to contain enzymes that are involved in germination. Could also contain enzymes. Oh, okay. So when it's time for them to rupture and start multiplication, so they have enzymes for that in the spore coat. Now we have cortex, which may occupy as much as half of the spore volume, rest beneath the spore coat. It is made of a peptidoglycan that is less cross-linked. Okay, cortex is also a layer. Okay, it's a layer and it is beneath the spore coat, and it is very thick, made up of peptidoglycan that is cross-linked than in vegetative cell, less cross-linked. And I don't know what it gives the property to the cortex when it is cross-linked, but they say it is cross-linked and obviously made up of peptidoglycan, because what other chemicals are they going to use for their cells? The spore cell wall or core wall is inside the cortex. What? The spore cell wall. Oh, okay, cell wall. How many layers are there? First is the exosporium, thin layer. The other one is the whatever it was, the thick layer, and the cortex, spore core. Then the cortex, which takes almost half of the size, and then we have spore cell wall. So it is already four layers. Okay, that's around the protoplast or spore core. Then we have a core. So the core has normal cell structure such as ribosome and nucleotide, but is metabolically inactive. Yeah, they are shut down, they are locked. It is still not known precisely why the endospore is so resistant to heat and other lethal agents. Now we still don't know how it is, because it is made up of peptidoglycan. Peptidoglycan is in the cell wall, is in the plasma, peptidoglycan is in the cell wall, of course, not the membrane. But why does this peptidoglycan layer and different complexity make it more resistant to heat and lethal agents than the normal ones? So as much as 15% of the spore's dry weight consists of the dipicolinic acid complex with a calcium ions, which is located in the core. It has long been thought that the dipoclonic acid was directly involved in resistant whatever. So they are saying that the spore, so the layer cortex is made from peptidoglycan, but the dry weight has How much? 15%. 15% is not that high, but it's not low. It's considerable amount. So dipoclonic acid with calcium ions, they are saying that it is located in a core, and they thought that it do what? That it do what? Come on, man. Don't stop in the mid sentence. I don't understand then. Come on. Where are you? Oh. Okay. Let's see. In heat resistance. Okay, so they thought dipoclonic acid is involved in a heat resistance, but heat resistant mutants lacking dipoclonic has been also isolated. So how does they play a role? Calcium does add in resistance to wet heat. Calcium, okay, so we have seen that the calcium add in resistance to wet heat. Wet heat means steam heat, okay, just as an autoclave. Oxidizing agents and sometimes dry heat. So they are saying that in heat kind of treatments or oxidizing type of treatments, calcium has shown the role. It may be that the calcium dipocholinate stabilizes the spore's nucleic acid. In addition, the specialized small acid soluble DNA. Okay, they are again using the word maybe. So maybe it is, but it's not proved, I guess. In addition, specialized small acid soluble DNA binding proteins are found in the endospores. Acid soluble DNA binding protein. Acid soluble DNA binding. Oh my goodness. DNA binding proteins are found in the endospores. Oh, okay, so they are talking about things except the calcium and the dipoclonic acid. I didn't know they were changing topic. By the way, we are moving forward, but I think we should see the diagram first, because why did they skip diagram like it was nothing? So first diagram. First diagram is, okay, we have said, that the what? When did this stop? Okay, wait, give me a second. Why is no one telling me that this thing stopped? Okay. Why did no one tell me that this thing was shut up? I should have noticed this one. Okay. So no, actually, why no one told me? Yeah. Whose fault is that my mouth was not moving from the centuries that I was talking? Should I be angry? That was not funny. I feel like it's been an hour since it closed its mouth. Like, for what other purpose does it exist? Whatever. It happens. It's life. So, again, the subterminal, the terminal. Subterminal means it's at the terminal but not completely. So, this is very close, this is less close, this is moving towards the center, and this one's very big. Even though it doesn't look like it is big, but it is extremely big, and of course taking up the cell of, material of space of the normal bacteria. Now there are layers. So we have studied the 4 layers. First one is an exosporium, and it was very thin layer. The other one was spore coat. So coat. It was made up of what? I forgot. Peptidoglycan? No. The cortex was made up of peptidoglycan and was not less cross-linked than the membrane. Then the core wall. So they are saying that the cortex takes up about half of the area. But aren't they lying? Like, it's not half. Huh? It's not half. 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 This is the cell. These are ribosomes and nucleus, and they are metabolically inactive. So okay, endospore structure, bacillus-centered endospore. Dipicholinic acid. Okay. First it is acid, so that means it has these functional groups HOOC, COOH, or whatever COOH is usually as nitrogen, and is it a benzene ring? But what is a benzene ring which has N attached to it? I don't know. It looks like a benzene type of ring. 1, 2, 3, 4, 5, 6. Okay, instead of one carbon, it has N. So the structure shows that it is very problematic structure. Just from the look of it. Also, I think this kind of structures are in nitrogenous bases as well. Okay, so now. Okay, let's see the how is it made. The diagram of the grand endospore. The father, the children, the resistant, the entity of the bacteria that leads it to next generation, the legacy that it carries, the hardship that it survives through. How is it made? So let's see the cycle. Let's see. What am I doing? Come on. So this is cell division. Cell division. We all know the cell divides. Yeah. So just so it is new. Then this is the wall. This is the DNA. This is the plasma membrane. So cell wall remains intact. Plasma membrane tries to break. So axial filament formation. Even though it's not filament, they are just saying that it makes loops type of stuff, dips kind of stuff. Septum formation and the 4 spore development. Septum means it septates, means it's partition kind of. Also there is a time linked to it. This is 4 hours. After 4 hours this thing is made. What do you call this thing? 4 spore. It is not a complete spore. It is before when it is a spore. Then engulfment of the endospore. See, here it is outside of the cell. It is like a bud. Then it engulfs. After it is engulfed, still not engulfed, it goes inside, and it has the same structure as wait, I see one thing. Do you see when it is engulfed, it also engulfed a part of plasma membrane of the original cell? So it kind of one plasma membrane, see here, one plasma membrane, and the other one that it originally had, the other plasma membrane, and then it digested some of the periplasmic space as well. Judging from the diagram, okay? Come on, move down. Don't cause trouble for me. Here, you see this area when it was engulfing, this area was engulfed with it as well, and this inner this pink area became a little bit creamy area, here. So if I am thinking correctly, this is the, let's see what it means. Also, the time, how much time did it took? 5.45 hours. So the 4 hours, when the 4 spore formation happening, it took 4 hours, while when it engulfed, it only took 1.5 hours. After it engulfed, the cortex formed. Cortex. Oh, so the periplasmic things and stuff here and there. So I mean, they might have been tightened. It matured. Let's say for simplicity, let's say it's matured. So it matured, and then OFM. What OFM? Oh my God! Outer formation. What is this? What is OFM? Do I look like an alien? Do they have abbreviation for OFM inner? No, F for 4 spore membrane. Okay, yeah, I'm not alien, man. Explain symbols to me. So inner 4 spore membrane, outer 4 spore membrane. Okay, the one that is engulfed before the membrane before engulfment, the membrane after engulfment, and this is, let's do cheating, spore coat, and the middle area become a spore coat. And this all takes, okay, after 1 more hour, from 5.5 to 6.5. I don't know what these blue things are though. What kind of changes are these happening though? But still, after 1 more hour, it matures and the cortex form. And the next is coat synthesis. Means, okay. I was wondering what these blue things are. Okay, so it's an exosporium. Even though it is thin, it is taking a lot of space. Give me a second. I need to see one thing. I need to see if my OBS is running or not. Yeah. So the next thing we see is exosporium formation. And then what happens to it? Exosporium means it is out of the spore. Then the supercoat forms. Okay, I think it didn't mature because it was waiting for the exosporium formation. So as soon as exosporium formation happened, the supercoat is a spore coat is formed as well, and it gives it refractability and heat resistance. What is refractability? Refractility. Okay, we will search what that is. So how much time did it took for that happen? From 8 hours to 10.4, means 9-10. So 2.5 hours for the further maturation, the exosporium formation, the spore coat formation, and then see, the endospore does come out. It's just endospore because it is from inside the cell. Almost a portion of the cell becomes a spore. Now when it is free, it looks something like this with 4 layers: exosporium, spore coat, cortex, and core. Is this even considered a layer? It's 3 layers. Then, okay, so we were thinking of searching for refractility. What am I doing? And how much did we cover? Or should I even read this? The thing that is below the diagram. Here. The stages are indicated by Roman numerals. The circle numbers in paragraph refers to the ors ends of the logarithmic phase. Logarithmic phase. A typical vegetative cell 4 stage cell. Septation 5.5, stage 3 self-engulfment. Man, we understand that, right? Why do we study that? Anyway, we were here. Now after we studied how the endospore is forming, now we are going to study the components of it, like what are the things that gives endospore the properties they have. So the thing that we will study is dipoloclonic acid and the calcium ion. I have so hard time reading this dipoloclonic acid. The next thing is that we have small acid soluble DNA binding protein. So the next thing that we are seeing is the proteins, okay, DNA binding proteins are found in endospore. They saturate spore DNA and protect it from heat and radiation, desiccation, and chemicals. So we know that DNA is wrapped around in the proteins, right? But in case of bacterial cell, it lacks a little bit of that, in comparison to our cells, eukaryotic cells, Yeah, because they are prokaryotic, they don't have that much machinery, proteins, materials, etc. in that. But in endospore case, they have proteins that make them compact, so it kind of protects them. Then I already said that we vacuum the water out of it. So the dehydration of the protoplast appears to be very important in heat resistance. Why? Because water has molecules and water has high heat capacity. Water absorbs heat in high temperature, so it's better to remove the thing that keeps the heat in. So we remove the water and we cause them dehydration. The cortex may osmotically remove the water from the protoplast, thereby protecting it from both heat and radiation damage. The spore cord also seems to protect against the enzymes and chemicals such as hydrogen peroxide. Okay. They are saying multiple things are protected by spore cord and cortex. Yeah. Also, they are saying the water that is removed is by cortex. Finally, spore contains some DNA repair enzyme. So they have, wow. So they have DNA repair enzymes for in case the injury occurs to DNA. But since if your DNA is damaged, it's like bacteria is gone. So the DNA is repaired when the spore germinates and the cell becomes active again. So it is repaired afterwards. In summary, endospore heat resistance probably is due to several factors. Okay, now what we all studied is summarized here, and we are studying almost heat resistance. So first is calcium dipicolinate, an acid-soluble protein, stabilization of the DNA, protoplast dehydration, the spore cord, the DNA repair, the great stability of the cell protein in the bacteria adapted to the growth at high temperatures and other. Still, it can survive under the 121 degrees centigrade. I mean, we can't either, okay? Endospore formation are called sporogenesis, and sporulation normally commences when the growth ceases due to the lack of nutrients. See, in case of fungi, the spores form under normal condition, but the sporogenesis or the sporulation means the endospore formation normally occurs when you see that I'm not growing even more and the nutrients are gone. So this is their last resort. It is a complex process and may be divided into 7 stages. So we already read that, so let's just read it for fun. Number 1. An axial filament of the nuclear material forms. Stage 1, following by inward folding of the cell membrane to closest part of the DNA to produce 4-spore septum. Stage 2, the membrane contains to grow and engulfs the immature endospore. Stage 3. What? First, axial filament forms. The second, it folds inward and a part of DNA is enclosed. The third, the membrane continues to grow and completely engulfs the immature endospore into a second membrane. So this is stage 3. Next, the cortex is laid down in a space between 2 membranes and both calcium and dipoclonic are accumulated. Okay. The cortex is laid down in space between. See, I told you when it was engulfing the blue part with it. So this blue part has the dipoclonic acid and the calcium in it, and it is a part of cortex. Even though the cortex is still not mature, it's just accumulated. Protein cores then formed by around the cortex. Protein cores then are formed. Okay, so the core is formed around the cortex, and the maturation of endospore occur in stage 6. Finally, latex enzyme destroy the sporangium. Yeah, these last stages are just summarized in one. This forms, and then enzymes kills this cell and come out of it. And then what happened? Finally, destroy. Sporulation requires about 10 hours in Bacillus megaterium. So this one, I think the one that is shown above is maybe Bacillus, or this is just general example. 10 hours is in Bacillus megaterium. Oh no, this is the life cycle of Bacillus megaterium. So it took 10 hours. Wait, did it or no? The last one was 10.5 hours. So yeah. Now how do you make these spores again into vegetative spores? This is an important part. So the transformation of the dormant spores into active vegetative cells seem almost as complex process as sporogenesis. It says seems, but it occurs in 3 stages: activation, germination, and outgrowth. Often a spore will not germinate successfully even in nutrient-rich unless it has been activated. So the activation is needed. So activation, germination, maturation. Wait, what? Maturation? Outgrowth. Outgrowth. Okay. This is shown as germination. Oh, guys, do you remember that how these plants germinate from the seed? Look, it's the same thing. This is seed and this is the cell that is germinating instead of plant. Yeah, so it's germinating. Oh, that prepares soil for germination and usually results from treatments like heating. So the activation So how do you activate it? So first is activation is process that prepares spores for germination and generally results from treatment like heating. So heating is a general activation process for that, and it is followed by germination, the breaking of the spores dormant state. So this process is characterized by the spore swelling, rupture, and absorption of the spore. I think it takes nutrients from the environment and starts accumulating in themselves, making water again and everything again. So the core part of this endospore is kind of swelling and it is having nutrients and water, etc. And it is activated by heating, so it kind of is ready after absorbing nutrients, etc. So how does that the inside part gets out, because it is covered by so many layers? It either by swelling, like I said, it absorbs quite nutrients maybe, but again it is it is not permeable. So how does it? Maybe due to heating, the pores are formed. Rupture or absorption of the supercore. It either absorbs the spore core or it either ruptures it. If it needs the spore core, it will absorb it. If it doesn't need it, it will rupture it. Loss of resistance to heat and other stresses, loss of refractility, and loss of spore components, and increase in metabolic activity. Just because it wants to survive and reproduce and have increased, this is actually metabolic activity. It loses a lot of things. It loses the spore components which were formed, the layers. It actually loses its expensive coatings. I don't know what this refractility is, but they lose this ability too. We have to search for it. And other they lose resistance to heat. They now can't be heat resistant. They lose quite a few 5-star points just for them to grow and survive again. I think it's a good thing because, who cares, because they can make endospore again because they are the same species. Many normal metabolites or nutrients, amino acids and sugar can trigger germination after activation. So now they are activated. So how does these two trigger the germination? Again, like I said, it is the growth. Like if you see nutrients you accumulating, and then you say, oh yes, we have nutrients outside and now I am safe, so it kind of triggers the germination that you can go out. The germination is followed by the third stage, the outgrowth. The spore protoplasm makes new components, emerges from the remains of the spore coat and develops again into an active bacterium. When it removes all the hard shells, all the coatings, etc., it can now act as normal membranes and cell walls, and it can absorb nutrients, etc., and then outgrowth. The growth starts to occur. So we have done it. We are done. So should we be happy? We are done with this chapter, and the next time, and I'm sticking to this style because it is more interesting to me, reading like this and recording how you learn new stuff, Even though I'm kind of surprised because wait, I didn't use Google too much, but we have to search one thing, refractility. I said I'll search it. Refers to the property of being highly refractive, means the spore appears bright and distinct when viewed under. Okay. Why does dehydrated unique chemical compound including the oh, okay. Okay. I see. They look invisible. They look shiny because of their structure. So guys, chapter 3 has been ended. Next time I will come with a new avatar. I plan to make a new avatar for every chapter, yeah. So next time will be chapter 4. Okay, so this one we studied prokaryotic cell structure, right? The next one will be eukaryotic cell structure and their function, and it will be with new avatar. You see this thing, where is it? Come on, get unlocked. Okay, you see this thing, this avatar is, this will be changed. You will see a different one. You guys can suggest to me how should I design it, or I will just design it myself. So yeah, expect a new this thing, new avatar, and yeah, a new chapter 2. And again I'll be using my voice because it's more understandable. And yeah, expect a lot of long videos because I'll go on and on. And I think I probably will start to stream more, because I think I was not doing it, probably because it was stressing for me, because probably it took a lot of stamina for me. Do you see how much time did it took for me to read just 3 pages? Like, this is not even funny, not funny. Yeah, should I start running now? Wow. No. Okay, what am I doing? So bye, see you next time. And yeah, good luck to everyone studying and things. Also comment, so I know someone is listening, because I see viewers watching, but I don't know if they are understanding a single word or not. So yeah, bye, good luck.